This is episode 1 of If I Can Explain It, I Probably Understand It. Today I'm going to explain DNA replication initiation in prokaryotes. So let's see if it actually makes sense or if I have just memorized it. Before going into the bigger picture of what goes on, why that happens and how it's happening, let's see the smaller picture first. So what exactly is DNA replication? What happens in DNA replication? What's the most layman version of it? Okay, so to explain this, let's say that let's take our prokaryotic OG cell, E. coli. Okay, so let's just say this ugly looking thing is the E. coli. Okay, now here, let's say the E. coli is actually a museum. Okay, and in this museum there are a lot of crap present, and one of such crap is the DNA. Okay, is the DNA. Now the DNA is your nothing else but a very high value manuscript. Okay, now if there is a museum, there is going to be a heist, right? So in this heist the key players are player A, B, C, and G. They are the key players. This heist, so we have the museum, we have the DNA. Now what? So we know that every heist movies, as we have seen, there is always the security is always very tight, right? But there is always one loose cannon. We're gonna exploit it. Okay, these heist players, they're gonna exploit that. So let's just say in the security of this manuscript, there is one particular guy in this one particular region. Let's just call that guy O. Okay, security guard O is pretty. Let's just say, for polite words, lax. Okay, these players, they know if they want to have a successful heist, they have to target our security guard O. Okay, so for that comes our first player, number 1, A. A is the charmer. Okay, so during the museum visitation, player A basically goes and charms the security guard. Okay, now the security guard is, while the security guard is distracted in the charms of beautiful A, uh, player B and remember, heist players B and C, these two basically go in here and take opportunity of security guard O's distraction and start uh, and and get this correct opening and start picking the locks to the secured manuscript. Okay, they pick the locks and kind of open the vault to get the manuscript. And then comes our player G. Once the lock is open, the vault is open, player G is the main guy, because okay, it's a heist, right? But if you completely take off the manuscript, it will cause more alarms, right? And there's a higher chance that you will be getting caught. So to avoid that, they are going to put a copy of this manuscript here. Now G is responsible to make this copy. So G basically, using this opening created by A and B and C, G basically makes the copy. So that was your DNA replication initiation in the most layman terms. So now let's see what actually happens. Let's zoom in and see the biology of the story. So let's zoom into the bigger picture. Let's zoom in what's the actual biology of the story. So our security guard O is nothing else but the infamous ORI. Okay, so what is ORI? ORI is the origin of replication. Okay, so this origin of replication is a very important component of the DNA, because without the origin of replication, neither the initiation will occur and neither the actual replication. So ORI is the weak spot in the security of DNA, if you will. How special are these two regions? How though? So the ORI is a 244 base pair long stretch of the DNA. Okay, so now in the most rightmost part, it is flanked by this four stretch of sequences where each stretch is like a 9 base pair long. Okay, so this region is known as the RRS, not yeah, the RRS, which is your replication, recognition, sequence. This is your actual weak spot. Okay, now, and the other region is your which is at the leftmost side of the ORI stretch. It is basically a 13 MER region. It consists of three stretch of sequences where each is like 13 base pair long. This region is known as the due element. Why it's called a due element, we will know that in the due time. Okay, that was a very lame joke, but I hope you have laughed. Okay, now what happens exactly, we will see that, but one important thing we need to know before we go into it is that both these regions, the due element and the RRS, both these regions, the due element and the RRS, they are both very AT rich. Why is this important? We will see soon. As you can see, the sequences, like the each sequence in the four stretch sequences is like the TT, AT, NC, and AT rich. Same with this. So moving on. So what actually happens here is quite simple if you understand the mechanism of it. So there is this protein or enzyme protein called DNA A protein. Okay, DNA A protein. This basically recognizes your RRS sequence. Okay, it recognizes your RRS sequence and it goes and binds to it. Okay, and as it binds slowly, not actually slowly in the bacteria, it is pretty fast, but yeah. So as it binds, other multiple DNA A proteins also come and bind your RRS. Okay, now as they are binding this RRS, as it's binding the RRS, what's happening is that this binding kind of creates a negative supercoil. So the DNA like coils and loops around like this. Because of this negative supercoiling, what happens is that there is some stress around the edges of this coil. Okay, now to explain that better, let's imagine there is a ribbon. Okay, you have taken this ribbon, pretty blue ribbon. Okay, satin one. Now what will happen if you like, you have also like fixed its ends. Okay, you have fixed its ends, because if you can like correlate, because this is ORI, let's just say it also has fixed ends, because it's not just a like segmented fragment, right? Fragmented segment. God, so it's not like that. It is, if you remember, attached to the rest of the DNA. So yeah, so you've taken the ribbon and it has fixed edges. Now what happens when you will loop this ribbon, as you can imagine, because the edges are fixed, there will be some stress and tension around these parts, right? That's the same thing that happens here. Now because of this stress induced because of the supercoiling, um, the stress travels to this region. What is this region? It's the due element, right? Now if you remember what is these regions sequences like, they are very AT rich. Now as they are very AT rich, what will happen? Okay, let's just see the difference between AT rich sequence and GC rich sequences. So if we take a sequence which is your AT rich, let's say AT A G C A T A, right? It will be like A T A G C G T A G. As you can see, adenine and thymine have two hydrogen bonds, while GC have your three hydrogen bonds. So obviously, your A and T have a less stronger bond comparative to your G and C, right? So and because of this, what happens is, even because of the slight stress towards this dual element, this will basically just kind of snap off. Like, these bonds will kind of snap off because of that stress. These hydrogen bonds, these hydrogen bonds break. And this will in turn result in formation of like this open complex kind of a structure. Like, this was your DNA, and it goes like this and this. I don't use the stylus a lot, so I'm really bad at using it basically. So bear with me. So yeah, this is the DNA element, and because of the stress created here, these have like their double bonds, not double bonds, what the fuck, these is your dsDNA. Here the hydrogen bonds have snapped. So because of this, there is this like open complex formation, the opening that we talked about before. So now this opening is very masterfully exploited by your player B and C, right? So who are these player B and C? Player B is nothing but a protein called DNA B. DNA B is your helicase of prokaryotes, basically. So okay, so we know helicase will unwind the DNA. So why is this player C needed? So basically, in your the DNA B in prokaryotes, especially E. coli, exists as a hexamer. So it's something like this. I hope that's not six. Yeah, it's six. It exists something like this. Okay. Now this DNA B, this DNA B. Okay, so why this open complex necessary? We saw that, yeah, this open complex was formed. Now why is this necessary? This open complex is necessary because without this open complex, we cannot load our DNA B here. Okay, we cannot load the helicase under the strands. So this open complex is kind of like a prerequisite for helicase working. So yeah, so now we know that our DNA B is actually a hexamer. Okay. uh, Now, yeah, okay, so we need C? So this DNA B, it cannot actually even now load onto this single strand here. Why? um it cannot load into the single strand here. It needs some help. So that help is our DNA C. So what happens is, DNA C exists as monomers. Okay, so there are six DNA C monomers. This is your DNA C. There are six DNA C monomers that kind of attach to this DNA B hexamer, and they basically stretch your DNA B. Okay, they stretch the helicase hexamer, so that this opening, this little thing, this opening kind of becomes a little bit bigger. The pore becomes a little bit bigger, kind of like, in more layman's terms. And because this becomes this is because we're able to like stretch this DNA B, we are able to easily slide it over this strand. Like, again, we have an analogy for this also. Imagine the DNA B is like those beads, bracelets that kids wear in the friendship, those beads friendship bands or something like that, or beads bracelets or something. So like, when you want to like get this bracelet over your hand, that's a very ugly hand, but yes, you want to get these bracelets over your hand, you basically kind like stretch it, right? Stretch it a little bit, and then it easily glides over your hand, and then it's there. So similar thing happens to load the DNA B. DNA C stretches your DNA B, and then it's easily able to glide over the stand, and it's loaded. Okay, so cool. We have loaded the DNA B. Now what? So we know DNA B is your helicase, right? So as a good helicase, DNA. So if we have loaded this, right? Now your DNA B is functioning. It's DNA B is unwinding. So we have loaded your DNA B, and DNA B is unwinding your the DNA from that open complex region, and this is kind of forming a replication bubble, if you will see that. Now we need, as we saw, there's another player, right? G will come here and copy this DNA. But before that, there is actually another player, an unappreciated player. Okay, this is called your SSBP, which is your single strand binding protein. So now this DNA, upon the unwinding, this DNA is like single stranded, right? So why? So the single strand poses two risks mostly. One of the risk is that because they are single stranded, these are the hydrogen bonds. These are kind of like sticky ends, right? Uh, to prevent, so there's high chance that these two ends, the single stranded DNA strands, will snap back together, right? And the other risk is that there is a high chance that the E. coli's inner machinery will degrade it, because it's single stranded DNA and it's technically not supposed to exist technically. So to avoid that, to avoid the degradation, our SSBP will come and join wherever there is a single strand present, like this, to avoid the degradation and the snapping back together, or with some, or like wrong snapping back. So that happens. Now at this moment, your DNA G is basically your DNA G. DNA G is nothing else but a primase enzyme. Okay, so this primase enzyme will come into the mix and form your RNA primer, which is again a prerequisite for the DNA polymerase to work. So it is an RNA primer. It will form, DNAG is your primase. It will form your RNA primer, which will provide the DNA polymerase with the 3' OH end, and polymerization will happen. So that was all about DNA replication in prokaryotes initiation part. Okay, that was so awkward, but yeah. Anyways, so that made more sense than I had expected. So I guess if you liked it, if you liked my chaotic revision and journey of figuring out if I actually understand what I read, shocker, right? So do think of subscribing. Thank you. Thanks for watching.